Hi,
Hi, John. How are you?
how are you? Not bad.
Good. Nice to see you.
Nice to see you.
How's life in New York these days?
Yeah. Well, COVID is real. I can tell you that.
My gosh, who knew that we would be so susceptible?
Yeah. Well, it's compounded by, well, anyway, politics aside, yeah,
Yeah, yeah. Isn't that a shame that we have a political piece that lops into all of that?
beautiful paper. Congratulations on acceptance.
Oh, I'm so excited.
Yeah.
It's nice to finally get it out. I actually tried to get it in a higher tier journal and they would not even review it.
Well, all of that is at some point is secondary, you know.
Like it. That's kind of where you get after a while.
I agree. I'm there.
I just
So,
get so burned out of it. I'm just kind of like, all right, I'm going to
yeah.
try a couple, but then I'm going to go to journals that I know will actually review the paper.
Yeah.
Yeah, yeah.
So what's the mechanism in the sperm again? What exactly is the
Well,
uh,
so it's a mitochondrial defect, so
yeah.
they can't. There's less of them, and they are less modal. But we haven't teased apart that mechanism in any more detail because funding ran out, and
Right.
so it could have probably been its own story if we'd have been able to chase that down. But I didn't pursue it further because of funding issues.
Mm,
Yeah, and so uh, I don't really know. I have a colleague at another place. Do you know Iqbal Hamza?
very well.
Yeah, so Iqbal's got a story he's working on that's about also a sperm defect that's heme related. And he kept bugging me, why aren't you doing this? Why aren't you doing this? I'm like, I can. He said, well, did you at least look at them by EM to see what the mitochondria look like? And I did not even do that because just
Hmm.
limitation of bodies and money and things. So I don't
Hmm.
really know the answer to that piece. We should check on your presentation. Have you done these
Oh,
Zooms before?
I have. So I should go to share screen, right?
Yeah. And
Uh,
your laser pointer, do you know how to make that not an arrow but brighter?
yeah. Hold on. Let me see if I can pull all this off. And do you see my
There it
laser
is.
pointer?
Perfect. Perfect.
Okay, that's great.
That's wonderful. Yeah. So anyway, I sort of expect, you never know
I
how many
actually see
people are on
the.
this, but we'll
Oh,
see. TK, are you on, I see?
yeah, I'm here. I'm actually seeing your presenter view instead of just the screen.
Oh, yep,
Uh,
I agree.
Oh,
Jonathan,
The
can you go to the slideshow rather than the presenter's view?
um, and slide display settings, swap presenter view. Is that it?
Yeah, that should be it. Perfect.
There
What
it is. Thanks, TK. I didn't even notice that.
What do you
Yep.
see on your screen right now?
Now I just see your presentation.
Okay, thanks so much. And then, and it works. Okay.
All right, that looks great. So TK is a graduate student in Adam Hughes' lab.
Hi.
Hi, everybody. Yeah, thanks for giving this talk. These have been awesome. It's been really cool meeting everybody from the iron community around the U.S. and around the world. It's been nice.
That's what I think.
Jer, are you on screen?
Well, I should be.
You are.
Can you see me?
Believe me.
I can't see me, but that's all right. That's not important.
How
There
are you?
go.
I'm doing well. How are you doing?
Usual.
How's life in New York?
Chaotic. Scary at times, or it was scary.
Yeah, I can imagine. I speak to my buddy Kushner every now and then.
Is he living in New York?
He is. He's on the east side, about, I don't know, 68th Street.
Yeah. It was not the right time to move to New York, I can tell you that.
Well, planning is everything. So after we hear you talk, we're going to go visit your buffalo on Antelope Island.
Oh, yeah?
Yeah.
Where is Antelope Island?
You remember, we took you there, we saw buffalo, and you told me the story about how they're all from the Bronx?
Yes, that's right. They are from the Bronx.
Yeah.
And I don't remember the story.
The Bronx Zoo had a colony, and
Here I am.
the buffalo were wiped out. Were wiped. I understand were wiped out from the plains, and they were transported. They were transplanted out from the Bronx Zoo. So.
I always
New York
thought they
Kings.
came from Neil Young's population.
I have no idea.
That may be a more recent version.
That's fun.
So, Jerry, how often are you at work?
Never.
Sounds like a good,
I've really retired.
really? Yeah,
Except that you're still reading grants for people.
there you go.
Occasionally.
Yeah. Well, that's great. That's wonderful.
So, John, how much are you working these days?
100%.
Are you still course master?
I do that. The coronavirus got me in the hospital for the past 2 months, and I'm sort of out of it now. Fortunately, I can get back to work, No one was prepared for this, and I've heard terms like black swan events and things like that applied to such an event. But I guess most of us survived. The patients didn't, but most of us survived.
Yeah.
It's a terrible thing it causes a myocarditis and as well as respiratory disease, and so people come in with a diffuse pneumonia and MIs at the same time, and there's nothing one can do. It's really a sad. I think everyone has post-traumatic stress disorder, really, to be honest.
Yeah.
So,
My friend Barbara Cahill essentially said the same thing. Patients come in and there's not a lot you can do.
no, but the system was not geared for anything like this. You see, if an emergency room only accommodates 75 people, what happens when 300 people show up in that shift? And I don't know what it's like in Utah. I think the numbers are lower there, but they're not going to be always low. They're going to grow. And
No, in fact, our state has not taken it seriously, and the lack of leadership is just abysmal. So they relaxed what restrictions they had, and what a big surprise, the number of cases doubled.
sure.
It's really quite sad.
Yeah, these people have no accountability, really. And but I guess who knows what's going to happen, and things may calm down. We can excuse people for lack of understanding, but at this point we can't do that anymore. Really, it's the absence of even a mask, a life-saving invention which cost $1 to make. We didn't have any masks back in March. We didn't have any, we couldn't find even masks to wear. So, the idea of going to work and sort of, into a tumult, which is your obligation, and everyone does it with great joy and everything, in retrospect, is crazy that leadership abrogates its responsibilities so readily and is off the hook so easily, is what I think.
Well, the fish rots from the head.
Yeah.
And clearly that's what's happened.
Yeah.
So is your wife still working?
Yeah, she is. It's mostly by Zoom, mostly outpatients at this point, and that's a nice thing, I guess. It's a little bit silly to, see patients on a flat screen rather than to examine them. But of course,
very surreal, actually.
very surreal. You know, the most important thing is that anyone over the age of 65, which is almost me, has to absolutely stay away from all of this. You know,
Yeah.
It becomes it's a lethal disease as you get older, and it's very important to be scrupulous in lack of contact with people, unfortunately.
As a result, we only see our grandchildren really by Zoom.
Right, that's right, yeah.
And your kids? Let's see, one was in college the last time we spoke. Has she graduated?
Thanks, she's in medical school. She's in my medical school.
Oh,
I
you
was
brought her into the business.
I was her first teacher. She squirmed like mad. And I said, you know, we had a conversation right before school started, and I said, you know, her name is Julia. I said, you know, Julia, not everyone likes me there. You know, some of the students will say, that guy's an asshole. What about that guy? He's miserable. He's a lousy teacher. He's awful. And she responded, you know, Dad, I'm a grown-up now, and we don't get embarrassed about our parents anymore.
Ooh, I like that quote. I may have to give it to my children.
Yeah, right.
Now, your son?
I have two daughters. The other daughter will head. Oh, there's Iqbal. The other
Hey,
daughter.
Sean.
Hey, how are you?
Good. Are you up and running?
Yeah, kind of. We fake it. We fake that we're doing coronavirus. So, you know, a little bit. My other daughter will be a lawyer one day. She will be suing police departments, that type of thing.
Got it.
We'll wait just one more minute here before we begin. Just give a couple more people a chance to join in.
Okay.
And then, John, I will introduce you just very briefly.
Okay.
And yeah. If everybody who is on will take their videos off and mute themselves, that would be great.
Right. Probably should do that myself.
Up to you, John.
Hi.
Meeting here. In the phone stuff.
Okay, we'll start in just 1 minute.
Okay. I probably should take my video off if I know how to do that. Oh, I'm not sure I know how to do that now.
You're just fine. It's not a big deal. We'd rather see you and your presentation, so no problem.
All right.
Okay, I'm going to get started. We want to welcome everybody to our 4th virtual seminar series for the Center for Iron and Heme Disorders. And today we're pleased to have Jonathan Barasch from Columbia going to give a talk to us. Jonathan got his bachelor's degree from Dartmouth College and then went on to get his MD and PhD from Columbia. Is that right?
Yeah.
Yeah, okay. And Jonathan is a physician living through the trenches of COVID in New York City currently, and has graciously offered to give us a talk on iron, cystitis, and pyelonephritis. And we'll turn the time over to you, John. Everybody, if you will mute yourselves and turn off your videos, then we'll have the best long distance interaction that we can. If you have questions, please type them into the chat. And John, I might interrupt you periodically if we have a question that seems like it needs to be asked at that time.
Alrighty.
Okay, thank you again for presenting.
All right. Thanks so much. And hi, everybody. I was once an iron biologist. Maybe, hopefully, again, I will become one shortly. My title is iron, cystitis, and pyelonephritis. First, the clinical problem of the
Let's see.
Does everyone hear me?
There we go. You're back up.
Okay. Let's see if I can get my laser again. First, does everyone see the laser pointer? Is that okay?
Yes.
Okay, first the clinical problem of this infection and the question of iron thievery. So here we go. The topic is the most common disease of the urogenital system. We call them UTIs. They are approximately 13% of women and 2% of men will have these infections every year in the US. The prevalence is about 50% of women will have one UTI during their lifetime. So this is the most common urogenital abnormality that there is. It's the most common kidney disease, the most common bladder disease. The effects of a UTI, we generally consider benign because there's a series of host defenses, but there are complications such as chronic cystitis, pyelonephritis, infection of the kidney, and then the same bacteria present in the bladder and in the blood system, I think by an unknown mechanism as to where the bacteria escape. Usually what happens, though, is that, and the reason it's not considered sort of a front burner issue is that the urinary tract has sufficient resources to expel the infection. First, the cells of the bladder die very quickly. The superficial cells are found in the urine with bacterial colonies associated with them. So there's a shedding of the surface of the bladder and recruitment of innate immune responses, including chemical responses. So usually these infections are dealt with pretty quickly, but on the other hand, it's not uncommon to talk about chronic cystitis, pyelonephritis, and urosepsis. So in the setting of the urinary tract, which has, the urine has sub-micromolar levels of iron in it normally, bacteria must obtain iron in order to replicate. And it's of a central concern to their biology. They produce a variety of specialized molecules known as siderophores, which are organic molecules that steal iron from our high affinity binding sites. This competition between the bacterial mechanisms of iron acquisition and host sites of sequestration has been called nutritional immunity, meaning that we carefully hold on to substances which bacteria would otherwise need. So in addition to expressing these organic molecules, bacteria also express receptors for them. And those receptors are often permeases and allow the recapture of these siderophores. For instance, FEP-A would be the receptor for this siderophore enterocheilin. Dr. Mobley has characterized these siderophores and their receptors and has proposed that antibodies against iron transporters make valuable antigens for bacteriostasis. In fact, there's some of the most highly expressed molecules, and the experiments are done by, which I've started to do, but Dr. Mobley has done the most of, introduce bacteria into the urinary system, recover them, and then examine either the selection of a certain subset of bacteria or RNA-seq to basically examine what types of proteins, what type of genes are upregulated. So siderophores come in many flavors and sizes and shapes. A large number of them have these dihydroxybenzene groups, which are the catechol groups. You can see here in bacillobactin. You can see enterobactin has the same group of molecules of these catechols, the three of them. You can see that some siderophores have two catechol groups. In our urine is a set of small catechols that are very abundant into the high micromolar range in our urine. which are derivatives of metabolism. And of course, not all iron chelation is due to hydroxyl groups, but nitrogens can also be involved. And of course, the most famous is the heme ring, which with nitrogen chelation. Some of these molecules, their oxygen interactions with iron is through the catechol ring, but in other cases, it's essentially a catechol, essentially carboxylic acids or ketone groups that are involved in chelation of iron. So they can be subdivided into the catecholates, which are predominantly these dihydroxybenzene groups, nitrogen donation of electrons to hydroxymates, carboxylic groups, and then there's a group that sort of are more difficult to classify because they seem to have multiple different types of functional groups. So when bacteria enter an iron deficient environment, and these are urinary bacteria we're looking at here, in vitro, in this case, exposed to two different iron deficient environments. You can see that many of their siderophore synthetic genes and receptors, there's FEP-A again, the receptor for anorexin, synthetic genes are upregulated. There are additional siderophore families that also undergo upregulation, and I'll call your attention to the CHU family here at the top. So bacteria upregulate siderophore expression upon iron deficiency, and apparently many of these genes are under common regulatory systems, for transcription factor. In addition to having the capacity to obtain iron by this upregulated system, bacteria cause damage to the host, sometimes by the expression of hemolysins. So in this case, we see after a urinary tract infection that the level of, this is non-heme iron, increases approximately 3 to 4-fold. And it does so within a few hours of the infection. So the entry of bacteria into our urinary system causes iron shedding into the urine. Upregulation of bacterial siderophore systems, shedding of iron into the urine as part of the process of, I assume, of bacterial growth. But even with these systems, one of the interesting findings is that one can manipulate the system with diet. So despite those systems I just mentioned, apparently it's not a saturated system. And one can supply iron to a mouse, for example, and the number of colony-forming units in the urine increases. Or one can place a mouse on an iron-deficient diet for a period of weeks in this case, and the amount of infection decreases. These are female mice that we generally use to create a urinary tract infection in the mouse. And I can tell you that it's a bit of a humiliating experience to create a UTI in a mouse, because what happens is that if you introduce the bacteria, the mouse essentially pees on you, and creating this variability in the data. So it's this type of spread of data is common in this field and is to be expected. So all experiments have to be n equals 10 as a single experiment. Recently, the NIH has, as you know, has asked everyone to have balanced experiments between males and females, and creating a UTI in a male to find the urethra in a male mouse is more than humiliating. It's butchery, essentially. So we've recently been able to. We visited a lab that does this routinely, and we figured out how to do it. So we're looking at male mice now as well. So the question is, where are the bacteria in the kidney? And I have to tell you that a scan of the literature shows not many answers. We understand it goes, the bacteria send through the ureter and enter into what's called the collecting duct system. But exactly where bacteria are over time is I'm studying right now. I'd like to know the anatomy of where the bacteria are. And I'd like to use different knockout mice, which I'll show you in a second, to examine the distribution of bacteria in the kidney. So my story in this starts many years ago, and I'm going to tell you about an important gene that becomes central to the type of work that I do. So here is I began in the 1990s with a part of the kidney. This is the primordium of the collecting duct. It's called the ureteric bud. It will form collecting ducts of the kidney. I made cells from them. I grew 25,000 flasks of these cells. I collected the conditioned media. I placed the conditioned media on heparin-cephros columns, and subsequent anion exchange columns, hydrophobic interaction columns, chromatofocusing gel filtration, and arrived at fractions that were pretty pure that contained molecules that, when placed on the primordium of the rest of the kidney, this is the origin of the collecting ducts. This mass of tissue is the origin of the more proximal part of the nephron, from the glomerulus up to the collecting duct. When one places the conditioned media from this source onto the recipient, which is called the mesenchyme, quote unquote, the mesenchyme, one sees tubulogenesis. So it's able to find molecules that make nephrons form. So cutting sections, we see a series of nephrons with a glomerulus and a tubule that are induced by these fractions. The response was so reproducible that one could use tubulogenesis as the assay to find active fractions. This particular set of experiments yielded LIF as the most likely inducing agent in this case. Pure LIF-induced nephrons. Obviously, this has to be a multi-step process. And of course, a single growth factor can induce many different events. But what we decided to do next was to examine the transcription factors that were induced over time in this process. So this is on day 0, and this is day 7 after exposure to LIF, for example. And what one sees is a wave of gene expression. And we decided to characterize the transcription factors that were induced. There are approximately 80 of them that are activated. And to think about what they do relative to tubulogenesis, we decided to clone all of them into adenovirus. Adenovirus is not a great means of gene transfer in embryonic tissue, in embryonic kidney, that is. They mostly infect stromal cells, but there is a small percentage that will infect the epithelial progenitors. And so we began infecting epithelial progenitors with all of these different transcription factors. The assumption is that more than one would be required to make tubules. And in writing a grant, which was rejected immediately because it can't be a single transcription factor, we thought about combinations and so on. Fortunately, most of the transcription factors did not induce nephrons, but this one did. And it induced nephrons exuberantly. So PAX2 doesn't do it, PAX8 doesn't do it, stats don't do it, et cetera, SMADs don't do it. But this gene, CP2L1, had this effect. The process was 100%, and feeling that we had made some mistake, maybe there was LIF present in the preparation, we sent the clone, we called the company and had them make a commercial version of this that worked like a charm. Add 10 to the 7 viral units, and the tissue would convert to epithelia. You can see the forming glomerulus here. In this tubule, here's what's called an S-shaped body. This is glomerulus, another glomerulus in red, and tubules in green. So this gene became of interest. And the story takes a turn, an unexpected turn, because What we thought we were looking at is the origin of kidney nephrons in particular, but the gene was expressed in parts of the nephron, but also in the collecting duct. And consequently, we sort of rethought the process. Here is this gene CP201. It's a cytoplasmic and nuclear early in development. That's embryonic day 13. This is the stalk of the future collecting ducts and the ureter, in fact. This is another stalk here. You can see later in development that the gene becomes a nuclear, located in the nucleus, and you can see that the stalk of the collecting ducts of the ureteric bud here has the gene, and the induced nephron has the gene. The part of the induced nephron is the distal convoluted tubule is this. The future limb Henle is this part over here. When we go to the adult, we find that the gene is distributed in the loop of Henle, the distal convoluted tubule here in red, yeah, and the collecting duct here along this line. So this is a gene that is expressed both in the nephron part of the kidney and the collecting duct part of the kidney, spanning these segments which are collectively called the distal nephron. The collecting duct is really interesting because what you see it as a series, it's not homogeneous like most other parts of the kidney. It seems to be quite heterogeneous in cell type. The blue cells are called intercalated cells. They're responsible for pumping acid into the urine and acidifying the urine. The other cells in red are called principal cells. They're responsible for recovering the final amount of sodium that's present in the urine to keep us in sodium balance, as the intercalated cell keeps us in acid-base balance. You can see CP201, this gene that we had found, is present in both cell types. If you look really carefully, there seems to be a third cell type. I'm going to point here, and I'm going to point here. These are cells with CP201, but they're not as blue as intercalated cells, and they're not as red as principal cells. We went earlier in development to figure out what was going on. And what we found is that early in development, the collecting duct, the ureteric bud and its branches forming the collecting duct system were very homogeneous. They all expressed keratinate and calbindin. But somewhere around E15, E16, E17, one begins to see the appearance of a different cell type, and marked by this ATPase and FOXI1, a transcription factor. These genes are very typical of future intercalated cells. But you can see that these genes overlap with keratinate and calbindin, which will become markers of principal cells. And so at this time in development, let's say, pick a number, E15, E16, one sees markers of intercalated cells overlapped with markers of future principal cells. And so we call these double positive cells after the idea of lymphocyte development, where cells can express multiple markers, but at later stages, only one class. So this seems to be a transitional cell, potentially a transitional cell. Although when we began to look at the adult, we found one here. It has the red marker of the intercalated cell. It has the blue marker of the principal cell here, and here, and here where you see the white asterisks. As opposed to canonical principal cells, those that traffic in sodium and potassium. Canonical intercalated cells, those that pump H+ into the urine. And so what you can see here is canonical images, but non-canonical images as well. And we decide to do a lot of lineage tracing and so on that I'll just briefly allude to in 1 second, because it seems that the adult has, should we say, a spectrum of cells. This becomes important in a minute. My colleague at the University of Pennsylvania, Caitlin Shostak, who's a world-class biologist, just a wonderful scientist, identified in a single cell analysis, she published this in Science, that there was a group of cells right here, these minority cells, number 8, she called them transitional cells. They are the double positive cells that I'm talking about. They are obviously a minority of cells in the kidney, but what she found in terms of their gene expression was something that we had found 2 years prior and had published on, that I'll show you now in a minute. So these cells exist in the adult, and her nomenclature of transitional cell is something that begs the question as to their function. Are they really progenitors, as they may have been a transitional state in the embryo, or are they really separate cells with separate function in the adult? And I think that's an area of future exploration for me. So in the next step, my computer is taking a pause. 1 minute. So much for Zoom. Microsoft PowerPoint is not responding.
Are you not advancing?
Yeah, it's not advancing. In fact, it crashed. 1 second. I'm going to reshare my screen, I hope. Okay, 1 minute. Sorry about that. Let's see if this works. Stop. My PowerPoint. Okay. All right. And how do I get back to screen share? 1 second. I'm not seeing the document. Hold on. Let me hit screen share again.
Just started. Yeah. Okay. Now it's showing your PowerPoint, but not the presentation.
Okay, 1
So
minute.
you want to go to the slideshow, I think.
Sorry about that. And does everyone see this now?
There you go.
Yes.
Super. Thanks so much. So we have these cells, transitional cells in the kidney that could be on the way to forming products of 2 different types, or they may have a unique function themselves. So our next step is we decided to knock this gene out, and as usual, no phenotype, unfortunately. We knocked out reflox exon 3, 4, and 5. Wasn't obvious by looking at the kidney. However, when we look carefully at the thick ascending limb of Henle, and when we looked at the collecting duct, there was an unmistakable phenotype. And that is that these blue cells, which are the intercalated cells, are gone almost completely in this knockout. You can see that the duct system that remains, this keratin 8 duct system, remember that was one of the original genes defining the ducts. Here and here, these are 2 different branches of the same duct system. You can see how ratty and irregular it looks. But the genes that are typical of the principal cells or the primordial collecting duct remain. But the diversity of cell types is gone after this knockout. The intercalated cells come in flavors, which I'm not going to talk about too much. One is alpha, which secretes protons. One is beta, which secretes bicarbonate. Both of them are gone in this knockout. The intercalated cell that disappears in the knockout, in the CP201 knockout, is composed of these, have these proton pumps, have carbonic anhydrase, which takes hydroxyl groups and turns them into bicarbonate. But what's interesting for this conversation is that they seem to form these omatidia structures. These pentagons are surrounded by the other cell type, the principal cells, over and over again, and the cells appear to spiral around the collecting duct, creating this sort of torque system. The green is CP201, which is found in both cell types, is highest in the intercalated cell. So if you put a spot imager on your confocal and go over these cells, what you basically find is that, you know, there are two canonical cell types. One is called intercalated cell, one is called principal cells. You can see that they have different patterns, amounts of gene expression, although nothing, we're surprised, nothing is pure here. There's always a mixture, a little bit of one thing versus the other. Then when you knock out CP201 and run your spot imager down the length of these ratty collecting ducts, what you find out is that there's really homogeneous staining, that the heterogeneity that you see in normals is gone. So what we decided to do next was to find out the genes that were downregulated in the knockout. We then went to CHIPS, we then chipped the DNA, and we did ChIP-seq, and we examined the genes that were knocked down, were they chippable in the kidney? And here we have to use whole kidney, and we can't use just collecting ducts. We probably should have done it in a cell line, in the cell line I talked about, but we did it in the whole kidney. We found that a lot of the knockout genes were chippable, therefore they are potentially, you know, possibly directly regulated by this transcription factor. So here's a list of the genes that are downregulated. And if we put them on a map, this is before single cell analysis, if we put them on a map, what we find out is that a lot of them are in the ureteric bud and cortical collecting duct and medullary collecting duct, and also thick limb of Henle. There's something called HLs here, somewhere here, that one would have assumed. So the genes that are downregulated check out as being chippable and check out as being located correctly. We took the downregulated genes and the next thing we decided to do was to ask, well, if we put these in a pathway analysis, of course, the word kidney should come up, the word loop of Henle should come up, collecting duct should come up. But the only pathway that really hit statistical significance was Notch. It turns out that intercalated cells make jagged one. Here's an intercalated cell. Here's an intercalated cell. And surrounding them in the principal cells is the activation of Notch. This is the signaling cell, the intercalated cell. The recipient cell, which has Notch 1 or Notch 2, and that's debated. We think Notch 1, other people say Notch 2. But around them is activated Hess, downstream of Notch. And when you knock out these cells completely, there's no more Notch signaling at all. And so then what we said is that if the intercalated cells are making jagged, to talk to Notch and the neighboring cells of this sort of omatidial structure, what for? And so we decided to knock out jagged. And rather than these very beautiful, discrete cell types, what you find is that the cells are all double positive cells all over again. In other words, all of the cells have blue ATPase and red keratinate, typical of this embryonic stage through which these cells had originated. All of this is to say there's a multi-step process of making these cells, but it's also a second important take-home message is that if you're going to study this area, there should be a note of caution in your studies that what we call intercalated cells and principal cells, sure, they are confocal. You can see them by confocal. You can rev up the contrast in your confocal. You can say I have a pure IC and a pure PC cell, but what it turns out most likely is that this is a spectrum of cells that are reflecting an embryonic past and a diversity of cell types, one of which is we're all going to call an IC, we're all going to call a PC, and we're all going to call a double positive. But this is obviously an area of examination. I'd like to point out, this is important, I'd like to point out that what I'll call intercalated cells for the rest of my talk I may not be the right nomenclature anymore. It could be that these so-called double positive or transitional cells, because they have a high percentage of the phenomenon that I'm going to talk about. I think that this is important because it could be that the cells that have responses that I think are of interest are not the canonical sort of ends of the spectrum type cells, IC and PC, but something in the middle, which Shostak would like to call transitional, and I'll stick with double positive for a moment. In fact, these cells are very metabolically active and label very quickly with RNA labeling tools that I'll describe right now. So in the collecting duct, we have a variety of cell types that are probably interconverting, and the exact location of genes of interest has to be rethought. In fact, every perturbation of the system, if a UTI, for example, affects jagged signaling, it's going to perturb this spectrum that I'm talking about. So it's very important to take a step back from the system and think about it. My favorite gene that comes from the collecting duct is called NGAL. NGAL is a gene that's expressed at very high intensity in collecting ducts. It's shown in red here by in situ in this human nephrectomy. It's also expressed in the limbs of Henle. And the issue is that it is CP201 controlled. And what you find here is that the blue is aquaporin marking the collecting duct, so it's very intensely expressed. It's not specifically an intercalated gene, which I thought in the mouse. The mouse and the human look different. It is all over the place. The red actually is in situ for NGAL, and the blue is in situ for the collecting duct marker, aquaporin, the principal cell marker. So here's an IC cell, no aquaporin. Here is a PC cell, aquaporin, and they both have different amounts of NGAL. So this is a robust limb Henle and collecting duct cell gene that disappears in the knockout of CP201. The mice, it's much clearer. It's collecting duct and thick limb of Henle, and it seems to be IC more than PC in this case. But again, we have to rethink these nomenclatures. Here's our luciferase mice, NGAL luciferase. If you tap on the kidney, the artery, if you give a nephrotoxin, the gene lights up. It's a medullary gene, goes down into the papilla of the kidney. The amount of it is dose responsive with injury. And if you knock out CP201 and end up with these collecting ducts that are pretty ratty, no intercalated cells, messed up collecting ducts, we don't see much NGAL available in the urine. So the collecting duct and the limb Henle make NGAL. CP201 regulates either directly or indirectly. And this became of interest because after working with Roland Strong, the identification of the fact that NGAL was a siderophore binding protein specifically, and the catechol groups fit into the pocket of Ent and trap iron. This is such a high affinity mechanism, and the location and the siderophore locates in the pocket at an affinity of 0.1 nanomolar. The complex is completely pH resistant down to a pH 4. In fact, you can wash this protein, a siderophore complex, for years. For years, I have things that are sitting around for years, and I keep washing them. They maintain the red color, typical of enterotelin being within the protein. In fact, it's essentially indestructible. So what we've decided to do as a proof of concept is we examine the structure of NGAL with Roland Strong. We produce a variety of mutations of the surface, changing positive charges to neutral charges or to negative charges. And that's important because what you can do then is you can inject this subcutaneously into a mouse, and it will appear into the urine a few minutes later. It will bypass all the systems in the kidney that tend to pick up positively charged molecules and lead them to recovery in the kidney. In fact, the more of this mutated NGAL, resurfaced NGAL compared to here, the more iron that you can drag into the urine. So this is pH resistant. It can, it's clearly enterochelan binding. It can bind to the small catecholates as well, or fragments of enterochelan. It can drag iron into urine and can actually move iron out of the body to some extent. If one gives 30 milligrams and so on this protein, one can see iron appearing in the urine, about a microgram per milligram of the compound, depending on preparation. So as a proof of principle, this is a very tightly bound system and difficult to peel off. This is the small catechol story. What's different about this story is that the catechols will also bind and trap iron in the calyx of NGAL, but it's very pH sensitive. So by pH 6.3, it decouples. So here we have a collecting duct with a spectrum of cells and probably a new cell type called a double positive cell, producing a variety of interesting proteins for the subject of potentially for iron defense, immune defense. Here we have an NGAL protein that binds to siderophores. And so the issue for me was to simply marry these ideas together. And the thing to marry them was a UTI. So if you inject bacteria into the bladder here, NGAL lights up in the kidney and the pelvis of the kidney. A tremendous amount of NGAL is made up in the kidney. And then for example, in the knockout of NGAL, one sees a persistence of bacteria in the urinary system. And I'd like to advance. And a persistence of bacteria lasts in the knockout. If one incubates the bacteria with NGAL or with desferoxamine, one activates gene, this is in vitro now, one activates gene expression along these siderophores. And so therefore, NGAL is a bacteriostatic agent that is specific to a class of siderophores that iron depletes the system. And these ideas were then coupled and confirmed by the fact that if you knock out CP201 and you're left with this ratty system, there's no NGAL in the urine. And in fact, there's log orders increases in bacteria throughout the kidney, the bladder, and the urine. The collecting duct is really important, and so NGAL is one of those of its products.
Jonathan, we have a question from Iqbal.
Yep.
Can K3-cis be used for iron overload or hemochromatosis, or does it not chelate cellular iron?
Yeah, so thanks, Iqbal. So quickly, the issue is that ENT is the most extraordinarily high affinity agent for iron chelation. It will rip iron off anything. So if you put transferrin with iron 55 attached to it, wash the transferrin 1,000,000 times, its content of iron 55 is pretty stable. If you put NGAL with enterochelan, white enterochelan, in other words, without iron, all of the iron will transfer from transferrin immediately. Within 30 minutes, you have complete transfer. So because K3 cysteine passes right into the urine, we can propose it as a iron chelation therapy. With the big caveat, and the gigantic caveat, is that we're talking about a protein that has a molecular weight of 20,000. So, which is going to be much larger than a clinically used chelator. But yes, in fact, if you give enough of it, you'll see iron depletion. And iron accumulation, the kidney and the urine, kidney, then the urine. You can do it radioactive iron, you can measure total iron. It's just a question of how much do you give. And so we were doing 30 milligrams a day for about 2 days or 3 days in a row, and we could see a flux of iron.
So can you tell if that's iron fluxing out of the cell, or is it just chelating the iron that's on transferrin in the serum?
Well, so in a hemochromatosis model, we see a change in pearl staining in the liver. So over time, this is going to iron deplete the system. Again, it's a large protein. In fact, if one had to think of a clinical use, I might not think of it as sort of a substitute for extrate or anything like that, but something that auxiliary to place in, like, for instance, old supplies of blood or something where there's liberation of iron. In any case, it's the TLR system that is being activated in the kidney that activates the expression of NGAL. If you knock out TLR4, you don't see much NGAL and you see much higher colony counts. Is this relevant to humans? And here I did a quick study of, again, we're frozen on the computer. Here I did a quick study of, I have to wait for the computer. Again, my apologies. Here it is. I did a study of students coming into the student health Columbia undergraduates. They all have UTIs, all these students, and you can see that there's a dose response between the amount of evidence for a UTI and the amount of NGAL that goes away on antibiotic treatment. So clearly we have a UTI response siderophore binding system from a source in the kidney that is abundant, that is very important for immune defense, because when you knock out the whole system, there is gigantic expression of colony-forming units. NGAL is very rapidly expressed. It's conserved in humans and mice. It's very specific to a limited form of iron chelation. Well, let's keep that in mind as I move on. So where are the bacteria? Where is pyelonephritis? And as I told you, the answer is, I don't think people actually have thought this out. I mean, obviously it comes up to collecting ducts, but then where are they? And where do they spread to? And what happens next? What's the natural history? And since there are many bacteria that have been mutated, and obviously many mice with relevant mutations in various iron systems, it would be wonderful to pair these and to actually map out bacterial systems. It turns out when you inject fluorescent bacteria, they go to a very particular place initially in the kidney, and they go up the collecting ducts, it's true. And these cells that they bind to, the green fluorescent bacteria, actually bind to intercalated cells. It's completely, unexpected, because it's the intercalated cells that pump acid into the urine, which serves as an antimicrobial. And it's these cells, or some spectrum of these cells, that is making NGAL in the first place. And while NGAL only binds to a class of siderophores, it just seems counterintuitive that these bacteria would go to the place that they're potentially limited in their growth. On the other hand, maybe the kidney uses this, and again, I'm frozen, my apologies. Perhaps the kidney uses this to trap bacteria and to either kill them or create bacteriostasis. So on seeing this, the story became quite weird. So here, for example, are bacteria binding directly to intercalated cells. We double color them so we're sure. They form colonies down further. They enter into cells into a compartment. I don't know what the compartment is. They escape from the collecting duct from intercalated cells. They glue themselves right to the outside of intercalated cells, like these bacteria right here. And they form larger colonies at different parts. And if you follow the bacteria infected, they are mostly in the collecting duct at this time point that we took. This is an 8-hour time point. They are mostly attached to inside or attached to intercalated cells, what we called intercalated cells, a spectrum of cells. That's much more abundant as a good chi-square here, because obviously the number of intercalated cells is much smaller than principal cells. So I felt that, the safety of the NGAL story, for whatever it's worth, I was leaving it because there's something much more complicated occurring here. So what I decided to do in brief is to, let me look at time. What I decided to do in brief was to create a mouse that had what's called UPRT. This is uracil phosphoribosyl transferase. This is a technique developed by Clary and Doe. I asked them for their clones. I recloned it into the Rosa locus. And so by using a Cree, one can activate UPRT. And what UPRT does is, essentially the mouse is normal and nothing much is happening, quote unquote, until you decide to inject 4-thiouracil. This UPRT is promiscuous enough that it can pick up 4-thiouracil and locate it into nascent RNA. So I thought this was a wonderful, non-destructive, you don't have to isolate cells, no fax machine, no single cell analysis, all of the complexity. If you have a CRE that's specific to a cell type, and again, it's CRE dependent and limited by CRE, but if you have that, you're able to label RNA at your time of choosing after an injury. And for things like UTIs and other types of tissue damage where genes are probably changing hour by hour, I thought this was a great tool. You simply inject 4-thiouracil, isolate RNA. The nascent RNA is thio-labeled, so you use thiobiotin and pull out the RNA and send it for sequencing. And yeah, you get 20-fold enrichment, depending on what CRE you choose to use of a given segment or of a given cell type. We've done tons and tons of pull downs from this, and I can say that it works great depending on your CRE and the abundance. Genes that are very specific, for example, aquaporin 2, if I can find it here, for example, can be pulled out with one CRE and not with the other CRE. Genes that are upstairs up in the proximal tubule of the glomerulus are not pulled out if you're using CREs that are down in the collecting duct. So this is a very, very simple system. And what we found from it was that in the kidney, we decided to take 2 time points. One is at 12 hours and one is at 24 hours. In retrospect, we should have done 8 hours, and at some point I need to go back. So everything I'm telling you is unpublished, and I've been holding it for reasons you'll see in a minute. We looked down this list and we did pathway analysis again of the genes that were expressed under the influence of UTI. And the gene list I came up was innate immunity, iron, heme, and the circadian clock are our top genes. And here's some of the players. So NPAS2 and BMAL are probably very well known to people who are listening. They're the circadian clock genes. And here's our friend Engel. This is Iqbal Hamza's great discovery. This is SLC48A1. So all of this is unpublished, and I'm wading my way through it, but we see that HRG1 is in vesicles. Yes, indeed, Iqbal. It's in vesicles in the cell, and depends on how you take the picture and where you look. It's in endosomes in these intercalated cells, and it also locates to the basal lateral system of these cells. Multiple antibodies do show this. The HRG1, or as Iqbal called it, or SLC48A1, is activated by UTI, as the pull down of RNA showed. The protein is pretty obvious. These are intercalated cells. So this is apparently a heme importer. What about heme metabolism? So here is HO in intercalated cells and in proximal tubules. Here's human that express HO on the setting of pyelonephritis. Here is a HO reporter mouse that when you inject heme, the kidney is magnificent. It lights up completely. And the product of heme is obviously biliverdin, bilirubin, iron, and carbon monoxide. So I asked my chairman to develop a probe for me for carbon monoxide. In the Chinese literature, we found this molecule. It's a palladium-based quenched fluor that carbon monoxide releases. And here's CO coming from the kidney, and that's from the liver. Kidney and liver. And so there's unmistakably a system here. When we do a UTI, we light up HO in the kidney. There's complexity in these mice that I don't have time. I can answer later if one wants to study the system. But UTI seems to do it. And to obviate the issues of reflux and getting bacteria to the kidney, what we decided to do was, in fact, to do this ex vivo. So we took a kidney, we injected urinary pathogenic bacteria into the kidney. We pre-incubated with an HO inhibitor, the famous zinc protoporphyrin. And this is the amount of carbon monoxide being released from the kidney in vitro. Infection induces heme cycling, heme breakdown, the release of carbon monoxide. It also induces, as our pull down showed, NPAS2 and BMAL seem to go up, which I read is involved in heme synthesis and in the circadian clock. So I'm wading into an area that I don't have any previous experience in. But I also understand they're heme exporters. They're very famous in red blood cells. It's FLVCR. The picture is unbelievable. So on the basal surface of intercalated cells, In fact, the alpha form, would be the beta form, it doesn't have it. In the alpha form, this molecule exists. All of this has been confirmed with RNA sequencing and single cell analysis more in the double positive cells than in canonical intercalated cells. There's a spectrum of cells, but perhaps this is actually functional in these cells. And it's very reproducible over and over and over again. So if we induce pyelonephritis, we see these genes, and so then what about the bacteria? So if you present bacteria with PBS, this is their growth rate. If you present them with biliverdin or bilirubin, this is their growth rate here on these curves. If you present them with very small amounts of heme, 1 micromolar, 2 micromolar, here's their growth rate in this green line and this pink line. If you use bacteria from Matt Mulvey, which is the CHU and HAMA knockout bacteria that have a deletion of heme transporters. There's some of the systems that I showed you are upregulated when you iron starve a bacteria. Then you get an intermediate line here when you present heme to them in this growth curve. And bacteria take up heme. There's a very large literature on this, although there are elements that have not been solved, especially, in fact, unfortunately for me, in E. coli, there's some steps of the process that are less well known than in other bacteria. Pseudomonas has been very well studied, but there's clearly heme oxygenase of some type, and there's carbon monoxide release here from bacteria when you present to heme in vitro. This probe does work. If you present it with NO, you don't get a signal. If you present it with these commercially available CO releasing probes, you can see a nice dose response. So the probe is working as advertised. If you take the CHU HAMA, which don't eat heme very well, and you compare it to wild type, and since these have different antibiotic resistance, you can double inject these, recover the bacteria, type them according to antibiotic resistance, and find out that in fact, CHU and HAMA, the mutations of the heme transporting systems in bacteria, gives you a deficit in growth, especially in the kidney, probably throughout the system, but it was most obvious in the kidney. And again, remember the humiliation of making a UTI in a mouse. So, this large spread of data is very typical of the field, is acceptable, but we think that there's a deficit, particularly in the kidney. And in fact, this is exactly what Dr. Mobley showed when he discovered the system. Carbon monoxide that is released, if bacteria are eating heme and releasing carbon monoxide, and mammalian cells are eating heme and releasing carbon monoxide, is that really what I'm wading deep into a thicket here that I'm unprepared for, but is it carbon monoxide that we really should be studying? After all, if you gas a plate of bacteria with CO, the bacteria do grow, but the colonies are extremely small. In the presence of the heme knockout system, of the importer system in bacteria, this is the famous chrome azole plates. The more yellow it is, the more siderophores the bacteria is releasing. It seems that there could be a toggle switch between the siderophore system and the heme uptake system, and bacteria can choose among them. But it turns out that in the presence of carbon monoxide release, all bets are off. Now, is that really what this is about? So in the final few minutes, what I'll tell you is that a bad event now occurred in my life, and a good event occurred simultaneously. The material that I just told you, all of this material is completely unpublished, and I'll tell you why in a second. I'm holding off for a specific reason. Well, for many reasons, as you probably can think through. But the NIH contacted us that our O'Brien Center would not be renewed, which supported all this research, if I continued to work in the kidney. And they felt that it's inappropriate for urologic grant to study the kidney. And no matter how many arguments I made, including send them a diagram of the kidney attached to the bladder and NGAL coming kidney and so on, the grant had to morph into a bladder grant. The RFA came out 6 weeks before the grant was due. So I assembled my lab of 3 people, and I said, we're going to just study the bladder. We ain't going to publish it. We have to study the bladder immediately. So this is probably the best thing that ever happened to me. You know, the timing of when you see bacterial colonies is very different. It's very, very early in the bladder. The entire urothelium, as it's called, folds apart completely after an infection. So there's a window of time to observe the kind of biology I'm interested in. Later on, the biology is a different thing. It's a matter of repair and chronicity, and it's a different question. Here are the intercalated cells back up in the kidney. It turns out that they express uroplakins. Not all of them, but some of them. And it turns out the bladder superficial cells here express uroplakins. And it turns out that our collecting duct system expresses CP201, which regulates the cell type. And it turns out that the bladder expresses CP201, which we don't know yet, but probably regulates the cell type. And it turns out that, I mean, all of this sort of happened like, you know, assembled it turns out that in amphibian and reptile, the intercalated cells are not in the kidney, but they're actually in the bladder. So the turtle bladder secretes acid, which is the function of the intercalated cells up in the collecting duct in the kidney. So by phylogeny and by sharing of some of these elements, we thought we could say something about the bladder awfully quickly. So thank God for all these databases that are out there. Well, all of the things I told you about FLVC, about HRG1, about HEMOX, about NGAL, is all discovered in the kidney. But what it seems to be is that the bladder has enormous expression of these genes, the superficial cells. I completely didn't know that. I missed the whole thing. And there's our friend CP201. So when you induce an infection in the bladder, all of the genes I just told you about seem to be in play as well. And if my slide would advance, here's NGAL after 4 hours of a UTI. Here is SLC48-1, which occupies endosomes of the superficial cells. It's in red. Green bacteria can be seen in these endosomes, subject of future research. Heme oxygenase is gigantically expressed on a UTI. The bladder, if you present it with some heme, pumps out carbon monoxide. If you infect the bladder, you get CO release. And finally, last data slide, if you do a UTI, the things that I found in the kidney with that RNA pull-down technique from the intercalated cells, the top gene was NPAS2 and BMAL, of the circadian clock fame of heme binding fame, are really upregulated likewise in the bladder cells as well. So I'd like to summarize the work I presented and to ask myself a bunch of questions about what I just showed you and where I need to go to bring these sort of phenomenological observations together. It's very clear that unless you capture iron, you're infected. There's no doubt about it. In fact, I asked an epidemiologist to tell me, of all the people taking iron tablets as an outpatient, who gets a UTI? Is it more common if you overdose yourself with iron tablets? And I think that's a wonderful study. We were not able to pull it off because of the background noise and a question of the timing of the iron tablet versus the UTI and so on. But I think one should do this study. The kidney makes a variety of proteins which have different spectrum of antimicrobial activity, NGAL being very specific to a class of siderophores, but the intercalated cells actually producing a lot of microbials. If you knock out the intercalated cells and affect the collecting duct, UTI is much more abundant. So the question here for the heme genes that I found in the intercalated cell, is this really a question of nutritional immunity, the kind of thing that we think about with siderophore capture seems like a much easier story. Is that really the case here? So one model is that this is about a competition for heme, the bacteria and their system, the mammalian cell and its system. This is the interstitium. This is the lumen where the urine is. And here is your docking site for bacteria, the uroplacans here. And on this side of the kidney, of these intercalated cells, on the basal side, we find in vesicles, and on the basal side, our friend, Iqbal's friend, HRG1, SLC48A1 here. We also find FLVCR, and if these genes are acting as importers into the cytoplasm, exporters, it's a curiosity that bacteria seem to locate at the site, or is it? Are they really here? Do they really locate to these cells in here in order to capture heme? Are these systems in competition? Perhaps FLVCR is active. I don't know. Maybe it's delivering heme to bacteria that are in this interstitial space immediately after the infection begins. So one idea is that this is sort of the old story of nutritional immunity. The second possible explanation for these data is that essentially it's true, and unrelated. I just happened upon the circadian clock. I just happened to find those genes, and sure, those genes are elevated in the UTI, but simply due to my enthusiasm, I see these heme systems and think that this is controlling bacterial growth in some way. But really what I'm doing is simply pushing up or down the circadian clock, which is a very prominent system in the collecting duct, has to do with water balance and other components. But,
Jonathan?
Sorry?
There's a question from Tom Gantz. Can you comment on endogenous siderophores and their role in a UTI?
Let me get to one sec. And so we have this heme system that I think, could be completely a figment of my imagination, that essentially I simply activated a system, and sure, we see carbon monoxide being released. On the other hand, perhaps the fact that NPAS2 and BMAL on the protein level are upregulated and produce, maybe they are driving heme synthesis, and HO is driving heme degradation, and perhaps the whole idea of the system is to generate carbon monoxide and antimicrobial. So, we'd like to establish, we think we see a pattern, but it could be true, and unrelated, I have to say. Carbon monoxide, of course, is the inhibitor of the system. So yes, we did stay in the lab all night long. We did do it. We infected at nighttime. We infected at daytime. There is an intrinsic rhythmicity. This is up in the kidney now. There is an intrinsic rhythmicity to some of these proteins, such as hemox. And a nighttime infection probably capitalizes on that and gives you a blast of hemox right over the endogenous rhythm. But this is all to say that maybe I'm just imagining these systems and pushing and pulling on a system. But I think the conclusion that I'd like to reach, and as sort of a hypothesis that brings together a lot of these data, is yes, it could be nutritional immunity. It could be carbon monoxide production. It could be true and unrelated. It's just the endogenous system. But what attracted me to thinking more about this as a physiologic mechanism is that the American Urologic Society, there it is, urologists say that there's 3 million red blood cells in the urine every day as normal. We normally have this microscopic hematuria. And my feeling is since every red blood cell is 1 billion iron atoms, and if you look up stuff in the literature and do a calculation, that could be enough to grow quite a number of bacteria. So in fact, the standard clinical marker of a UTI is actually hematuria. So in conclusion to the data, I think what we found is a physiologic system of heme capture and deactivation. that may supply a circadian clock. But what I'm imagining is that that system could be in competition with bacteria for heme. I'm going to hold the publication until I have adequate knockouts. The knockout that I chose so far was a floxed hemox knockout that I graciously Anupam Agrawal gave to me. The problem is we can't knock the gene out completely. We can reduce its RNA level to about 60%. Because we're dealing with many cell types, we had to use the Rosa-Cree. It was a Rosa-Cree to knock out in many cell types. And my feeling was that with a 60% knockout, we should look for a phenotype, but it's not going to be the answer. We have found a phenotype that I'm not showing you because it's just simply too preliminary. But you know, it could be that the location of bacteria is different. It could be the case. I've turned to my friend Iqbal Hamza, who has very graciously sent us the hemoxygenase knockout mice. We may have to cross them into mice to allow them to survive into adulthood. And he's graciously allowed me to take a look at the SLC48 knockout, because unless we have a knockout to give us whether the system is first relevant, second protective, or third pathogenic, I won't be able to conclude from the kidney and the bladder, which I'm going to publish simultaneously, whether I'm looking at things that are true and unrelated, or actually a form of what's called nutritional immunity.
Jonathan, I'm going to just interrupt you. I'm afraid we'll time out here. So I want to make sure that we get any questions that are going to come up.
And thank you very much.
Tom asked the question about commenting on endogenous siderophores and their role in a UTI.
Yeah, so the catechol system is present in the urine. It will dock iron into the NGAL, we can find that in the urine. We can do a natural product chemistry isolation of things that bind to NGAL and iron, and we always come back to the simple catechols. We do this in an unbiased fashion. I think what probably initially NGAL could be docked with those molecules, but then as one proceeds down the collecting duct to greater and greater acidification, that will fall off, freeing NGAL for enterochelan, would be my guess. I think that they are sort of a transient buffer higher up in the nephron where the pH is more favorable to them.
Okay, any other questions? Okay, Jonathan, thank you so much for your presentation. Really beautiful. The microscopy is incredible. Good colors. It's like the zebrafish system, so that's very nice to see. Here's one more question. Would it be possible to show iron accumulation in the urinary tract by using EM?
In the past, what we've done is we've loaded NGAL with radioactive iron, the enterochelan with radioactive iron. We have injected that into the blood system and also subcutaneously. We can find radioactive decays in the proximal tubule where most proteins that are filtered are taken up, except for K3 cysteine, which bypasses and goes into the urine, dragging iron with it. So where we see the radioactivity ending up. So I've used a radioactive approach. In chronic kidney disease, the kidney becomes Pearl's positive. So there's an accumulation of iron around sort of where the proximal tubule and the limbs of Henle organize at the cortical medullary junction. One can see an accumulation of iron in chronic diseases of where many proteins are passing the proximal tubule because of the chronic disease. But for experimental approaches, radioactivity is how we follow where iron goes.
Thank you. Any other questions? Okay, we'll say thanks again to Jonathan, and our next presentation isn't until the middle toward the end of July, but want to thank everybody for their participation and stay safe and healthy. And I'll end the meeting now.
Thanks so much, Diane, for having me.
Absolutely. Thanks,
nice-
Jonathan.
Nice to see everybody.
Nice
Bye.
to see you. Bye.
Bye.
See you, Jer.
See you. Oh, hold on. See you, Diane. Bye, Jonathan.
Bye, Jerry. I'll write you an email.
Yeah, please. Let's talk.
Nice to see you.
Stay well.
Okay, bye.
Bye.
Okay. Bye.